Welcome to Sage Green, where we delve into the wonderful interdisciplinary field of bioengineering. Today, we'll explore how the body reacts to the implantation of a biomaterial. I will cover cell adhesion and communication, biomaterial testing, and the effects on the body after implantation, including the immune response. So, to investigate biomaterial interactions, we first need to understand cell adhesion. You might have been taught that tissue is made up of its constituent cells, but you might not have known that the mesh that holds the cells together makes up the majority of the mass of the tissues in the body. The extracellular matrix, or the ECM, is composed of fibrous proteins such as collagen and elastin, in the gel of proteoglycans, which are polysaccharide chains linked to proteins. An important protein in the ECM for cell adhesion is fibronectin. It has multiple binding sites, as well as binding to the cell surface for adhesion between the cell and the ECM. Cells also adhere to each other and to the biomaterial where the ECM would have been if it is present. Zooming in even further, this is done in cell junctions with cell adhesion molecules, or CAMs. Due to the range in proteins involved and type of junction connections, there are a variety of ways a biomaterial can adhere to living cells. This can be used to our advantage by engineering biomaterials to express certain adhesion, for example, by controlling hydrophobicity and surface charge. Some properties can also lead to a change in other body responses. For example, the stiffness of a biomaterial affects the cell migration and differentiation, leading to a more integrated implant. This is when another important structural element comes into play, the cytoskeleton. The cytoskeleton is a complex network of protein filaments inside the cell that provides structural support and facilitates cellular movement. Actin filaments, microtubules, and intermediate filaments are key elements of the cytoskeleton. During cell adhesion, these filaments undergo dynamic rearrangements, influencing the cell's shape and behavior, aiding in cell adhesion, among other cell processes. Another important cell process that impacts biomaterial interaction is cell communication. Cells signal by excreting a molecule from one cell to the other, whose signal is then received and transduced throughout the receiving cell, leading to an eventual response. When designing a biomaterial, you first need to take into consideration the molecules that are going to be excreted when they come in contact with it, as they have different potencies and lifespans. For example, the nervous system is a very precise mechanism, so neurotransmitters have a low potency, which means that only a little bit of the neurotransmitter is needed to transfer the signal. There are also different methods of cell signaling in relation to the cell that need to be accounted for. After the molecule has been secreted, it can bind to the receptor of the plasma membrane, which can recognize either ions, enzymes, or other molecules, which can be recognized by either ions, enzymes, or molecules that bind to a specific protein. This is followed by a pathway leading to an eventual cellular response, of which proteins are often turned on or off. Cells are also able to signal more precisely and efficiently by combining signal molecules, using one molecule to signal to many receptors, and by changing receptor expression. There are many different signaling molecules, but the most common ones are the most important for biomaterial interaction are cytokines, and more specifically interleukins. Cytokines are proteins and signaling molecules for the immune system, and interleukins are a class of cytokines that mediate communication between leukocytes, which is a type of white blood cell. Their role in the immune response is vital to understanding transport rejection. For example, cytokines are released to activate the major histocompatibility complex, or MHC, molecules that in turn express intracellular antigens to notice and destroy foreign cells, which is useful when we get infected, but not necessarily when we want a material to stay after the transplant. Cytokines also regulate inflammation and immune system cell differentiation, among other functions within homeostasis. Because of their role in the immune system and inflammation, hypersensitivity and overexpression can lead to conditions like autoimmune disorders, rheumatoid arthritis, and cancer. However, our understanding of these signal molecules can also allow us to creatively solve these conditions and maybe reduce the chance of transplant rejection. Because of cellular processes, including cell adhesion and communication that we just discussed, care must be put into testing a biomaterial before clinical use. Biocompatibility is tested first via a risk assessment, then an in vitro test, and an in vivo test. During the risk assessment, the factors that influence toxicity are taken into account. The specific dose, duration, and frequency of exposure make negative effects range vastly. For biomaterials, the material itself, the site of application, and surrounding biology also have an effect. More specifically, xenobiotics, which are foreign molecules, can have chemical, biological, and physical effects to the surrounding cells. Even further, xenobiotics from implants can have systemic effects, which are those that travel to the rest of the body. For example, if a material is in contact with the brain, it can affect the whole nervous system. In vitro tests for biocompatibility are regulated under ISO and are designed to test for negative effects on cell function, and can also test for toxicity linked to a gene mutation if necessary. For example, in the MTT assay, cells' metabolic activity is measured after direct contact with the material. This is done by mixing MTT salt with cells that have been left to multiply on materials in an assay structure. Metabolic activity can then be calculated with a spectrograph, as the cells that have active mitochondria will convert the salt to a pink color. There are many other in vitro tests that can be used based on the application of the material. After, in vivo tests are carried out, often in small animals. For example, the intracutaneous irritation test tests for symptoms of inflammation after injection of molecules. As more innovative synthetic materials are being researched right now, there are challenges in studying long-term effects as well as obtaining accurate results while reducing animal testing.